Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Niao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 134th seminar, and I call it a Northwestern Day. I am in Korea, traveling since August 16, originally planning to give 6 invited talks at 2 conferences and multiple universities, including DTU in Denmark and Chalmers University of Technology in Sweden. Unfortunately, life is something full of surprises, and my dad passed away on August 17, and I am in his office room at midnight Korea after the funeral. He had been a healthy man until he found out my suffering caused by Washington University in St. Louis, leadership people's discrimination, harassment, and retaliation. I miraculously recovered from the 4 years of dying years, literally, but my healthy dad became sick, and now he is not with us. He was my role model, and also he will be with me forever because of his lifelong mentoring and teaching me, and I miss so much you, my dad. And I'm sorry, to start with this seminar, but I will overcome the pain, and then you will be with me forever. And thank you for your guidance until now and then in the future. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Keith Teo. He is a professor at Northwestern University. I have known him since 2005 when I joined MIT as a graduate student. As I mentioned to him recently, when I met him again in Dublin, Ireland, he was so kind to make me relaxed just before the ACS conference in Boston, where I presented an oral talk as a graduate student for the first time. For me, it was the first oral presentation at our international conference, and a simple, kind word meant a lot to me. And Keith has also contributed to the scientific community and education with his kind heart, and we thank him for his such effort. And I would say the most important thing in the world is not just power or money or something else, just simple kind word, or just sharing their time, or empathy, or just some resource, even some simple thing. And I believe giving is the real power of the change. And Kiss basically showed that to me many years ago. So Keith, thanks so much for your kindness and contribution to the society and community, and the virtual podium is all yours now, and thank you again.
Well, thank you, Taesuk. And while I've never met your father, I know you well enough to know that he certainly did do a great job, and has been a big impact on the world just through you. So Just want to make that comment, and yeah, just thank you for the super kind words that when you shared that with me last month. That made my day, just to know that some of those things matter sometimes. So I just want to give a quick talk. I'm not going to include any data. I really just want to talk about a particular challenge that I think biotechnology and synthetic biology is well positioned to make an impact in, and hopefully motivate some of the people that watch this talk into start thinking more about some of these questions. And this is an area you think about a lot as well. And so yeah, hope this is interesting to people and inspiring in certain ways. And it's about trash to treasure and converting pollutants and waste into industrial chemicals. And I want to start with just this slide about what microbes do well, because I think when we think about synthetic biology, we're often trying to solve some societal problem, biomedical problem. And the first thing we should probably ask is biology the right avenue to solve such a question? And I make this slide just to think about what are things that microbes, sort of the land that I live, do well. And I think one thing to think about here, right, concentrating dilute feedstocks. This is, you could think of this as separations in a chemical engineering setting. But what we have here are the concentrations of glucose and ammonium that E. coli can grow on and can effectively take up these molecules to supply itself with growth at these levels. And if you compare this to glutamate, which is an amino acid that has both carbons from glucose as well as nitrogen from ammonium, it's at 96 millimolar. That's a tenth of a mole is the intracellular concentration. And so if you look at this in terms of a separations process, you're getting 5,000 to 8,000 fold concentration of these metabolites is what cells are able to do, because they, of course, many of them live in the environment and have to deal with these very scarce resources. And so if you think about this, comparing it to other membrane or other type of separations technology, this is incredible that microbes can do that. And the other thing that microbes do really well is they do chemical reactions on wet heterogeneous feedstocks. And traditional thermo or chemocatalysis often doesn't do well with heterogeneous feedstocks, so it tends to work better with a more homogeneous feedstock in terms of controlling it. And then often things need to be completely dewatered in order to do a lot of the chemistry that we practice at scale. So this is kind of an important aspect of thinking about this. And so these are just two things that help guide me anyway, thinking about what are problems that microbes might be well suited to address. And so things where this feedstock is very dilute is something it does well, and things where the situation where it's wet and heterogeneous are things that cells will do well. And I've become very interested in the past several years about wastewater treatment. And I would argue this is the largest use of microbial biotech on Earth. In Chicago, where Mark and I are both from, the wastewater treatment plants here process 1.3 billion gallons of water per day, taking out different pollutants and providing clean water. They do this, it's a somewhat energy intensive process. So roughly 2% of the global energy goes just to wastewater treatment. And of course, there are lots of places in the world where proper wastewater treatment isn't happening. And then the carbon, phosphorus, and nitrogen are able to make it into watersheds, which is, of course, bad for the environment, the ecosystem, and ultimately humans as well. And so taking advantage of the biological processes that happen here is something that I think there's a real opportunity. Right now, the carbon goes to the CO2, because the goal is just to get clean water. So any way you can get the carbon out of the water, they're happy with. The phosphorus ends up in a compound called struvite, which is typically recovered for fertilizer type applications, and nitrogen gets converted into dinitrogen, which is what 70% of our air is made of. And so what we'd like to do is valorize the waste carbon and nitrogen in much of the same way that the phosphorus is becoming valorized right now, in terms of turning it into something that has a market and can be a value to society. And so really the goal of my research program is to think about what co-products could be made in this wastewater treatment environment that prevent waste from polluting the environment, but then also can we make things that offset the need for virgin products, meaning things made typically from oil, petrochemical, or I'll just have one slide on nitrogen, which comes from the Haber-Bosch process, which is also a very energy intensive process. And yeah, I love this quote. This is a chef in London who runs a restaurant that has zero waste. And he has this quote that waste is a failure of the imagination. I'll skip over this slide, but if you want to pause it to look at some of the details, the big point here is waste is good for an economic process because it's often negative value. So people will pay you to take some of this stuff off your hands. Certainly wastewater treatment costs something. So if you can offset that cost, then you can get that cost. And there's a lot of energy embodied in at least the carbon in different waste streams. And so it would offset up to whatever that is, a little less than 20% of the fuel that we need in this country. And so we could go pretty far towards making things like sustainable aviation fuel if we could efficiently recover these feedstocks. And in particular, nitrogen waste, which is what I've been working on for the past several years. These TG is a 10 to the 6th or a million metric tons. So this is the amount of nitrogen that is either being treated or is being used. And I'll be the first to say the biggest problem with nitrogen is non-point source sort of farm fertilizer application, which we don't directly address, but there's a lot of nitrogen that ends up in point source wastewater. And so that is an opportunity for us to be able to make an impact on. And I'll close on this slide, just saying our dream is to take something that looks like this. This is the current state of the art, where nitrogen on the order of 50 mg per liter gets treated to get it down to 1 to 10 mg per liter, which is sort of the EPA levels that are acceptable. And that nitrogen is currently getting converted into N2, which is essentially worthless. If it's run improperly, you actually get N2O, which is a potent greenhouse gas. And if you don't get treatment, then you get all the problems on this side. And so what we're trying to do is make a compound called cyanophycin, which is a nitrogen storage polymer. And we could use it, we could chemically modify it to make things like nylon or polymethyl methacrylate, which is the absorbent polymer in diapers. So we think this is a promising area of research to work in. And yeah, with that, I'll just show a quick slide. A lot of this work now is in collaboration with George Wells in environmental engineering, and Jennifer Dunn, who does techno-economic analysis and life cycle analysis, and has been a huge asset in helping inform our strategy. So I'll stop there.
Fantastic talk. I mean, very important area, as you can imagine. So waste is so much problem, global problem, and I absolutely agree with you. And I love, your one quote. I mean, the waste is the failure of the imagination. I absolutely love that. I mean, that means, our society already failed because we generating too much, waste last several, decades. So we need to fix that problem as soon as possible. So thank you so much for your wonderful talk. Thank you.
My pleasure.
All right, so now the main speaker of today with our longer introduction, Mimee is an assistant professor in microbiology and the School of Molecular Engineering at the University of Chicago. He is a 2022 Beckman Young Investigator, and I believe I wrote a recommendation letter for him, and this is one of the easiest letter I can write. His interest in microbial life began in Montreal, Canada, where he completed his Bachelor of Science in Microbiology and Immunology at McGill University, one of the best universities in Canada, and inspired by the nascent field of synthetic biology, Mark pursued studies at the Massachusetts Institute of Technology, or MIT, completing his PhD in microbiology with Dr. Tim Liu as an HHMI International Student Fellow and also Qualcomm Innovation Fellow. So he was already kind of rising star when he started PhD or later in his career. Interestingly, all three of us also today are MIT alums, so I probably call today MIT Day. Since starting at University of Chicago in 2019, his group focuses on developing strategies to precisely engineer the activity and composition of microbiota using synthetic biology and synthetic ecology approaches. His long-term vision is to implement these technologies to chart new basic and translational studies to exploit the microbiota for human health. He is truly a rising star, and I admire his work, especially the work he published with Tim Liu at MIT. Importantly, he became a father, I think recently, trying to balance work and family life. And Mark, thanks so much for your time today, and please take it away, and all yours now. Thank you.
Great. Thanks, Tae-Sok. I mean, first off, my deepest condolences for the news about your father. That's really tough. And I think, you know, even you being here, which I presume it's like midnight in Korea, like sort of just shows your support for young faculty. I mean, I felt that since starting here, like you had mentioned, you had written a lovely recommendation letter, and sort of throughout my career, it's just really nice knowing that there's sort of senior professors in the community that are looking out for us young guys that are trying to, you know, scrap by. But with that, I'll share my screen and start my talk. Let me just orient myself. Cool. So yeah, so like Tae-Sok said, my name's Mark. Our group is interested in engineering host-associated microbiomes, and I'll sort of tell you two kind of vignettes, two stories, some of the earlier stories from our group, and you know, would be happy to take and discuss more over questions. So to start off with, for those that are uninitiated, I'm going to be talking about the microbiota, or the microbiome, which refers to this sort of collection of microbes that live on and in the body. And there's a lot to say about the details of the microbiome. I teach a whole course on it, but sort of, you know, distilling it down into 3 words. The microbiome is exceedingly diverse. So within our body, we have hundreds of different species that occupy different spatial niches. We have different microbiomes in our skin, in our gut, in our nasopharynx, etc. It's extremely heterogeneous. So different people have their own microbiome, almost as unique as our fingerprints. Obviously, there's shared functions and shared species between individuals, but everyone's microbiota is different, and it also changes over time. And it's also complex. So it's not just microbes that are living in the body, but they interact with one another, they interact with the host, and they interact with our diet and our environment and these sorts of things. In terms of the composition of the microbiota, usually the lion's share of the research has gone into the bacterial portion, which play a major role, particularly in the gut, in linking the microbiome to host physiology. But of course, there are also archaea there are fungi, there are protists, there are viruses, there are worms that can occupy the microbiome as well. And the last piece that I'll say is that there was really a sort of renaissance in microbiome research that was catalyzed by advances in sequencing technology. So born out of the Human Genome Project, where the short read sequencing technology that after sequencing the human genome was sort of quickly adapted to profiling microbial ecology, at first in the ocean, but then it shifted over into the host. And it was really that sort of advance in technology that got people really excited about how microbiomes were different in different conditions. So this happened kind of in the mid-2000s, this sort of renaissance in research. And kind of over the next decade or so, what we more or less learned was that disease induces changes in the microbiome. And that is intentionally vague, because it really seems to be really every single disease that you can think of, from cancer to a variety of infections, to metabolic diseases, to immunological diseases, even some neurodegenerative diseases have microbiome signals. And maybe that makes sense because in a disease state, you'll presume that the physiology or the physiochemical environment that exists within a person will change, and that'll select for different microbes that are adapting to those different changing conditions. So that sort of first, era or renewed era, let's say, in microbiome research really sort of focused on this cataloging of microbiomes using sequencing technologies. And as we've sort of moved forward, a lot of folks are more interested in perhaps this more relevant question of not what does the microbiota look like, but rather does the microbiota affect disease? And there are definitely some cases where the microbiome is actively interacting with the host and either has positive or negative effects in disease states. But there are also others where it's merely just a correlation, where the disease state changes the host. This selects for different microbes, but the microbiome doesn't really have anything to do with those diseases. But even though this is a very simple question, right now it's often very difficult to answer it. And at least I would argue that's because of this technology gap that exists in making targeted perturbations to microbial communities. And these are just simple things, simple gain or loss of function studies, adding a microbe, subtracting a microbe, adding a gene, taking out a gene. And at least in my opinion, these sort of studies are really pivotal to understanding the mechanism and the molecular mechanism through which microbes interact with the host, so that we could really learn if and how they interact with the host in a variety of these different disease states that I have here. And as Tae-Sok mentioned, sort of my synthetic biology hat, which has sort of fallen off over the years, if I put it back on, what I'm really excited about is if we can engineer the microbiome to treat disease. So for these diseases in which the microbiome is important, these same sort of tools that we could use to make targeted perturbations to microbial communities, maybe we could use these same strategies to edit communities in a beneficial way to protect against disease or to treat the diseases. So what my group works on largely is what I call these sort of additive and subtractive interventions with the goal of restoring gut function. So, in this model, community, on the left, you have a dysbiotic microbiome. So that's consisted of strains that might be deleterious for health. And the overall goal is to restore it to the more green-oriented community that we have on the right, which is beneficial microbes that have the normal metabolic functions that keep homeostasis within the body. And here today, I'll talk just two little vignettes of sort of approaches, these additive interventions, which you can think of as sort of classic synthetic biology, trying to take genes and gene circuits, put them into bacteria, deploy those into the microbiome, so the microbiome now has a new function that might restore health. And on the flip side of that, subtractive interventions, particularly using narrow spectrum antimicrobials and more specifically phage, so that we can edit communities to remove bad players that might be pathobionts in the case of disease. So the first story that I'll talk about is Jack's project. He was the first graduate student that sort of joined my lab. Incidentally, also a Northwestern graduate, worked with Julius Lux there. So the Chicago connection is very strong on this call. But the general idea of what we were looking at the outset is that if we started off looking at genetic engineering efforts in sort of gut microbiome research and kind of noticed that they tended to be relegated to lowly abundant taxa that exist within the microbiome. So on the left is this kind of stacked bar plot that you see in a lot of these microbiome studies that reflects kind of the diversity at various taxonomic levels of different sorts of microbes, bacteria that would exist within the microbiome. And if you look at synthetic biology efforts, there has been a lot of examples in E. coli, particularly a probiotic strain called E. coli Nissl, engineering it for a variety of diseases. Here's a classic example from a company called Synlogic, where they engineered it to treat phenylketonuria, which is a genetic disease that causes the accumulation of phenylalanine in the bloodstream, and this can be neurotoxic. Here, they engineered E. coli to degrade that kind of excess phenylalanine to protect mice and later humans against PKU. So this made it all the way to phase 3 trials, and it kind of failed there, but is kind of maybe the most advanced example of kind of using synthetic biology for human health. Going back, I guess now 24 years, the first example of this was the seminal paper in Science in the year 2000, where they were engineering lactic acid bacteria, in this case Lactococcus lactis, to produce a mammalian cytokine, IL-10, which is anti-inflammatory, to treat inflammatory disease models in these mice. And kind of later on, there's been many folks that have worked on either Lactococcus or different Lactobacillus species to express and secrete proteins that can be protective against disease. And finally, there's some more recent work out of my group when I was back in Tim's lab, as well as sort of Justin Sonnenberg's lab, and Hirschan and Sirk, showing that you can engineer Bacteroides species, which are these teal ones on the left that are a little bit more abundant, to secrete nanobodies that could be potentially therapeutic. But kind of what we were interested at the beginning is, what about these more abundant taxa? So these beige taxa, these Lachnospiraceae, these Ruminococcaceae, these are dominant members of the microbiome, and they actually turn out to be quite important. So just a brief orientation of Lachnospiraceae. Here's kind of an example of a bunch of different species that are there. But within the family, these are gram-positive bugs that are obligate anaerobes. So they can only grow in the absence of oxygen, and they're exquisitely sensitive to even a single molecule of oxygen hitting the cells. It can cause kind of immediate death. So for these reasons, they're particularly difficult to culture and work with in the lab. But over the years, people have learned that they produce a series of unique metabolites with a variety of therapeutic capacity. So many of these species within Lachnospiraceae produce butyrate, which is a short-chain fatty acid kind of product of microbial fermentation, which, if anyone in the microbiome field can agree on anything, this is a beneficial molecule. It could make colonocytes happy, improve barrier integrity, and also induce regulatory T cells, which can kind of dampen inflammatory responses in the gut. But aside of that, there are specific strains of Lachnospiraceae like Ruminococcus navis or Clostridium scindens that can act on bile acids within the gut to produce immunomodulatory secondary bile acids that can either create beneficial immune responses or confer colonization resistance against pathogens. And then there are yet other species that in models of food allergy can by themselves kind of protect a microbiome or a mouse, in developing food allergy. So for these reasons, we think that Lachnospiraceae are a particularly exciting taxa within the gut microbiome, but there's kind of a lack of studies that have really delved into the molecular mechanisms through which they interact with the host. And that's partially because they're currently challenging to engineer. So really, Jack's project at the outset was seeing whether we can engineer systems within Lachnospiraceae so that we can expand their therapeutic potential. We know that they already have these native inherent beneficial effects, but can we engineer them either with additional functions or to shore up some of their shortcomings, for instance, their oxygen sensitivity and things like that? So I'm going to cut out a fair amount of, work and failure that Jack performed over the years in order to get things to work. But what we did is we started off with kind of shuttle vectors or plasmid systems that we really just borrowed from other sort of Clostridia communities. So there's a fairly large industrial Clostridium community that engineers Clostridia for a variety of purposes, degrading feedstocks into kind of value-added products and those sorts of things, as well as the pathogenesis communities, particularly for Clostridioides difficile and Clostridium perfringens, that have develop genetic systems in those organisms. So we use those as kind of the basis for engineering these Lachnospiraceae. We did extensive protocol optimization that was more or less based on the idea of using conjugation to introduce DNA into Lachnospiraceae using E. coli as a donor. And secondly, we developed these plasmid systems, which were great, but eventually our applications would be in the gut, where we would eventually deploy these engineered microbes, and we would hope that they would exist in a microbiome for a long period of time, and their functionality would remain stable. So that requires chromosomal integration, and we sort of leverage these type 2 intron-based kind of landing pads so that we can insert landing pads in the genome and use well-described integrases so that we can introduce DNA stably within those genomes. And also note too that there's kind of in the sort of start and stop of this project, there was kind of a similar work that was published in Cell in the year 2022 that worked with similar organisms, not quite the same ones that I'm working with here, but using this same kind of ethos and technique to engineer them. So to start off with, we wanted to kind of be able to express genes both constitutively and inducibly. So what we did is we started by taking promoters that drove predicted housekeeping genes within Lachnospiraceae, put them into our vectors, into E. coli, and mated them into our Lachnospiraceae recipients. And in kind of screening through a variety of these different promoters, we could put the reporter gene nanoluc, which is a luciferase that works well in kind of these anaerobic species, and introduce them into Lachnospiraceae, which at least at the time was the first that anyone had done this before. So this is in the species Ruminococcus navis, but we took the strongest promoter that we had there and showed that it could work across multiple different multiple different Lachnospiraceae species, including both butyrate producers, ones that are associated with health, ones that are associated with disease, and all those sorts of things. So Lachnospiraceae being kind of a fairly untrodden zone in terms of synthetic biology, we started off with these kind of ad hoc approaches in trying to introduce promoters. Since then, we've had a phenomenal undergrad in the lab that has kind of adapted some protocols out of Harris Wang's lab to do kind of high throughput assessment of transcriptional activity within Lachnospiraceae. So there, what we did is we synthesized oligo pools with all of the promoters in a variety of different species of Lachnospiraceae, cloned them into vectors, and created promoter libraries so that we can, simultaneously scan their transcriptional activity there. As you can imagine, these, this is like a library of thousands of different bugs. So we needed to use a technique where we would barcode the transcripts of nanoluc reporter, and then we could use amplicon-based sequencing and sort of normalize it to the DNA within our pool to see whether or not to look at transcriptional activity there. So this is some later data that we had. We had a successful first pilot pool of around 60 promoters or so. And, scanning through these different promoters, we can see that we can find promoters that span a variety of different orders of magnitude. So right now we're kind of in this scale up phase to get that 60 up to around 6,000. Lots of matings, lots of plates, so that we can, one, get this data, and two, try to model the transcription within these organisms. I'll say so far it seems to be fairly similar to other described gram-positives, which in some regard is nice, but we're hoping to see maybe they have some unique transcriptional signals there. So aside from that, we wanted to create inducible systems. Luckily, we were able just to port over the tried and true TET-R system and introduce it into our vector. And here in multiple species of Lachnospiraceae, when you introduce the small molecule ATC to induce gene expression, we could see across these multiple different species that we can get quite robust controlled gene expression there. Awesome. So the next thing that Jack was interested in doing was really creating a therapeutic. And the sort of easiest ideas that we had were to try to secrete mammalian cytokines from Lachnospiraceae that can kind of synergize with their inherent beneficial activity. So the first step of that was identifying kind of secretion tags that we can append to heterologous proteins that we would introduce into Lachnospiraceae so that they can secrete them into the supernatant, showing that they can make them, and then showing that they are bioactive in a variety of different respects. So to identify these sequence tags, we kind of used a two-pronged approach. So the first was kind of unbiased proteomics, taking supernatants from several different Lachnospiraceae species, looking at which proteins were enriched in their supernatants in sort of steady state conditions, and identifying and flagging those as proteins that might be good candidates that might have sort of amino acid sequences that would allow for secretion there. And we paired that with predictive modeling, particularly using signal P, which at the time we didn't know would be robust within Lachnospiraceae, but it turns out that it is, and use those to sort of figure out what kind of secretion signals that we could use to append to a heterologous protein. So we kind of fused it to our favorite reporter nanoluc, in this case under a TET-R system, and then we looked at the secretion to the supernatant. So on the left here is kind of the sort of baseline secretion or really excretion of nanoluke that you would see within a culture of Coprococcus combs, which is one of these butyrate-producing Lachnospiraceae. We get some leakage outside of the cell. But if you kind of append these secretion tags, you can see that we could vastly increase the secretion, at least at varying levels. On the bottom is the percentage of protein that is secreted by these organisms. And with some of these secretion tags, we can get almost 100% secretion. And with one of these fun things that I saw, what we thought was maybe a fun but potentially dumb idea is we just took the predicted secretion tags across Lachnospiraceae, made a consensus sequence that doesn't exist in nature, and fused that to our nanoluke. That's denoted here by this synthetic one. So here, just kind of taking residues that don't exist in nature but should kind of afford protein secretion, seems to do pretty well. It secretes all of the protein from the cell. So we thought that was kind of interesting. So next, we wanted to kind of secrete a payload that we thought might be therapeutic. So here we kind of identified this protein IL-22, which is a mammalian cytokine that in a variety of different studies has been shown to promote barrier integrity, sort of gut health, protect against a variety of different gut pathogens, as well as colitis and those sorts of things. So here we took the mammalian gene, codon optimized it, threw it into these secretion vectors, and lo and behold, we see that in the wild type cells, it doesn't produce a mammalian cytokine, which is expected and nice. But when we introduce it into, in this case, a plasmid system and add our ATC, we can get very high levels of IL-22 secretion. For reference, around 10 nanograms per mil is sort of the bioactive range. So we're exceeding that by 10 to 20 fold. So next, we did a bunch of other, bioactivity work that I'm going to glaze over here. But some sort of hot off the presses things that we got in the past month or so is seeing whether or not we can see some bioactivity of this Coprococcus combs produced IL-22 in the mouse gut. So here, what we did is we colonized mice with our engineered strain of Lachnospiraceae that natively produces butyrate, which promotes barrier integrity, like I had mentioned. And then we had engineered it to produce this secreted IL-22, which we hope would translocate across the gut barrier to signal to its receptor on the opposite side and hopefully lead to improved gut health. So one of the things that, is kind of one of the major downstream effectors of IL-22 signaling is the production of these antimicrobial peptides, reg3 beta and reg3 gamma. So in this case, we were happy to see that in the definitely in the ileum and to some extent in the colon, when we introduced our engineered strain of Combs, but not a wild type strain, we can see induction of reg3 beta and reg3 gamma expression here. So this is at least pointing to the fact that we're making some IL-22 that is causing some effect within the mice. And kind of the next step that we're going here is kind of going into disease models to see whether or not this IL-22 signaling is sufficient to protect against either colitis or sort of metabolic disease within these mice. So that's the first story, and I'm going to have a huge shift of gears to talk next about bacteriophage and sort of the idea of using subtractive interventions to remove bad players from the microbiome. So this is the work of the staff scientist Ella, who also formerly worked at Northwestern to maintain the strong Chicago theme that we have going on here, and trying to use these phages to kill pathogens in that respect. So at least at the outset of the project, this was very simple what the idea was. So these phages are viruses that can infect and kill bacteria, and they have exquisite specificity. So that is their upside, is that with most antibiotics, if you would use them, these broad spectrum antibiotics, they not only kill pathogens, but sort of also wipe out the beneficial microbiome as well. So we were excited in using phages since they tend to be particularly strain specific as kind of a surgical agent to sort of remove specific strains from the microbiome. So in this little cartoon that we have here, if you wanted to get rid of the orange strains, you can use your orange phage. If you want to get rid of your beige strains, you can use the beige phage. So this is relatively simple. We'll see that it gets a lot more complicated in the end. So we needed a target at first. So our initial target was this bacterium Klebsiella pneumoniae, which is a particularly scary multi-drug resistant pathogen that is of excessive clinical concern. So it's part of this group of organisms called carbapenem-resistant Enterobacteriaceae. This includes E. coli, Klebsiella, and Enterobacter species, but these are of particular kind of threat because of their high levels of antibiotic resistance, including some strains of Klebsiella for which there are no clinically available antibiotics that can treat. So for patients that contract these kind of pan-resistant infections, all that you can really do is provide supportive care and cross your fingers that the immune system can clear it. So we thought that this would be kind of a good starting point for creating a novel antimicrobial because there's a great sort of clinical need there. So as a little bit of background, so these are gram-negative organisms. They're highly related to E. coli, which is nice for a variety of reasons. And their kind of main virulence factor is the fact that they're encapsulated. So I often describe these as kind of a meat and potatoes pathogen. They don't have any of these fancy virulence factors or these sorts of things. The reason why they can cause devastating infections is because they're capsulated. They have O antigens. They can get iron quite efficiently. They're sort of very high on the defense and very low on the offense in terms of the pathogen. Like I mentioned, they're extremely antibiotic resistant. They're also particularly environmentally resilient. So when they get into hospitals, they're difficult to clear with a lot of sterilization techniques. And why we were kind of looping back to the gut microbiome, the sort of model for many Klebsiella infections is that healthy people can be colonized from their gut with Klebsiella pneumoniae without causing any issue whatsoever. But when someone's admitted to the hospital or there's some sort of error in their health, what can happen is that these bacteria can escape the gut and then translocate to other sites where they cause a bona fide infection. So this could be causing pneumonia, like their name suggests, urinary tract infections, bacteremia, liver abscesses, etc. It's really a Swiss Army knife pathogen that can cause a lot of issues, but kind of the issue more or less stems from the fact that they're in the gut microbiome. So the overall goal of the project, right, was to see, okay, well, can we get phages that can target Klebsiella? So this was kind of the basic sort of overall view of what we had. So we started off saying, okay, well, first we would like to create this phage bank. So to start off with, again, connection, previous speaker, going to wastewater, which is sort of the natural source of phages. So as you well know, these gut pathogens get flushed down the toilet all of the time. And there's many phages that exist within wastewater that can kind of target those. So for many of these enteric pathogens, using wastewater or sewage is our primary source of isolating these phages. And what we would do is we would have a strain of Klebsiella, we would have a sample of sewage, and we would either, enrich, filter out the dead bacteria, and spot these onto lawns of plates where you can get these zones of clearing or plaques that would be reflective of a phage. And there we would passage them, streak purify them, isolate these phages, and then, we would have phages that we can use to kill Klebsiella. So here's just sort of a sample of the kind of library of phages that we built. This is probably around a year old or so, but it looks pretty. So we have a variety of different strains of Klebsiella pneumoniae, quasi-pneumonia, and varicola. These include highly drug-resistant strains, as well as primary clinical isolates that we get from our clinic here at the University of Chicago. And they're relatively diverse. They have a diverse set of capsule and O antigen types. And in terms of our phages, here's around 50 or so that we have. Now we're up to 100 different phages that, span a variety of different taxonomy for phages. Could go into more detail, but we'll just say for this point in time that we have a lot of phages. And so with this kind of collection of phages, the question then becomes, okay, well, how do you prioritize phages? If you come in with a pathogen that you would like to target, how do you assemble the most optimal cocktails that we could kill those phages? So in order to do that at first, we needed to learn some things about these phages. So in data that I won't talk about, we did some extensive T and seq screens to look at host factors that were necessary for phage infection. The idea that if we had functionally diverse phages that kind of overtake different systems within the bacteria, then we can create a cocktail of phages that would be robust to bacterial evolving resistance. And here's an example of that here. So if here's a strain, our favorite strain really of Klebsiella, this MH258, which is one of these carbapenem resistant strains. Here we can select a variety of different phages that are taxonomically diverse that are all fairly effective at killing the Klebsiella. And when we combine these different phages and look at how good they are at suppressing bacterial growth, so on the left, this is normalized to 100% in the absence of phages. If you add a phage and wait 24 hours and see how much growth you got, this is what the data is representing. You can see that individual phages can suppress growth, but not extensively. However, when you use increasingly more complex combinations of phages, you can get more suppression of growth. You could also see that some phages are better than others. For instance, in these two phage cocktails, if you have phage 12 with any other phage other than 46, then you can get fairly robust suppression. But when you have 46 alone, it looks very similar to the single phage cocktail. And this is a good example because phage 12 and 46 share the same receptors. So they kind of are targeting the same systems in the bacteria. So it's the same strategies to evolve resistance there. So we could do this, with our bank, fairly readily with arbitrary strains of Klebsiella. So like I had mentioned, this is our favorite strain where we can take a cocktail and mix it with bacterial culture and suppress kind of the outgrowth of the Klebsiella. You can see with single phages, you get outgrowth at around 4 to 8 hours. You could do this with multiple strains of Klebsiella, just kind of mining through the bank, looking at their host range, looking at their taxonomic diversity as well as their functional diversity, and put together cocktails that can suppress the growth without any resistance that's there. Next, for one of the things that we did, at least with some of these cocktails that I thought might be worth mentioning, is including sort of an adaptive evolution step in the assembly of our optimized cocktails. And this is because when we're constructing our phage bank, we have strains of Klebsiella, we're isolating phages on those, and presumably those phages are best adapted to that strain that was the isolation host. But if you were to get a strain from the clinic, even if you have a phage that is able to infect it, that phage might not be in its sort of optimized genomic state, so to optimize its virulence so that it can be used as an antimicrobial. So there, what we can do is kind of sequentially passage phage lysates on ancestral strains of bacteria to kind of adapt these phages so that they can increase their virulence. And one of the examples here is with one of these phages, Phi-13. Within the dotted line is the ancestral phage, and at the equivalent MOI. With our evolved phage, you can see that it can kill the Klebsiella strains much more efficaciously. And if we sequence the phage, we see that maps to receptor binding proteins. And the reason why it can kind of infect more efficiently is because it can bind to the cells a little bit better, by order of magnitude better. So the last thing that I'll talk about in the final minutes is kind of the use of these phages in mice. And to sort of put the cart before the horse, it's a lot more complex than we thought it was. And I think it's also a good example, at least for me, coming from a synthetic biology field, we're very sort of a design and kind of goal focused, where we wanted to design a phage cocktail that would kill Klebsiella. It was a very singular and simple goal. But really, the system is a lot more complex than we thought it was. So what would turned out initially being as sort of negative data has really spawned multiple different research programs in my group to, one, both learn more about the basic biology of phages in the gut, and two, coming up with engineering strategies so that we can overcome some of these shortcomings. Anyways, so to get to the data itself. So what we thought to do was to use a gnotobiotic mouse model with Klebsiella in the microbiome to use it with phages. The reason why we wanted to do that is because most of the other sort of work in the field had been using antibiotic treated mice, where if you give Klebsiella to these mice, they take up 100% of the microbiome, which isn't really sort of the carriage model that we were interested in here. So here we had kind of a designed microbiome of 14 bacteria, on top of which we can add Klebsiella, where we would colonize and kind of engraft within its microbiome at around 1 to 2%, which is what you would see in that case of sort of pathogen escape from the gut. So within these mice, if you treated them with phage, we can see a modest but statistically significant and reproducible decrease in the amount of Klebsiella that we can recover in the gut upon phage administration. And we can see that this correlates quite well with how much phage that we're getting to the distal gut, so that if when we collect feces and titer the phages there, we can see that the kind of decrease in Klebsiella that we can get is nicely correlated to the amount of phage that we can get or detect in the feces. So this, again, points to the fact, which really other groups are working on, that formulation is a major issue in these kind of gut applications in the phage. Kind of improving the bioavailability of phage might make for a more effective product. Other things that we saw, which were quite curious, is that as opposed to in our in vitro context, where we would infect a culture of phage, it would select for resistant mutants. These would be genetically resistant, and then the phages would go away. Here in the gut, we can see coexistence between virulent phage and susceptible bacteria. So that there are some phages, even sort of, a week post-phage administration, that can coexist sort of with the bacteria in the gut environment, sort of suggesting that there is something with the physiochemical environment within the gut that is different than what we're getting in a mixed test tube. You'll also see that the kind of efficacy of the phages in replicating in the gut varies greatly from phage to phage, with this purple phage 39 being able to replicate quite well in the gut, whereas this phage 46 being able to replicate poorly and actually not at all. And if you contrast this to sort of our in vitro results, we really get this reverse in the efficaciousness of phage. In vitro on the right, you can see Phi-46 is quite good at suppressing phage populations, whereas the purple phage 39 is one of the worst ones, but this is inverted in the gut. So this is one of the things that we're really interested in now, to say, okay, well, clearly the environment has a significant effect on the efficacy of phages, but how do you define that? How can you create model systems so that we can model things in the lab? So, and then when we deploy them in the gut, we can predict better whether or not our phages would actually be useful. Cool. And in the final minute or two, I'll just kind of, mention the sort of maybe the final punchline of this work. So what we saw is that even though we got this modest decrease in the amount of Klebsiella that we can recover in the gut, that the bacterial populations changed over time. So, and they changed in the way that they would lose their major virulence factor, which is the capsule. So if you would recover bacterial colonies from these mice a week post-administration, we could see in some mice literally all of the Klebsiella had converted to an acapsular population, which is an avirulent population that can cause disease. We decided to look into this a little bit more and saw that, okay, well, we figured that maybe the phages that use capsule as a receptor were the ones that were selecting for this resistance. In this case, it's this yellow phage, phage 12, as well as this orange phage of 46. And if we measure the percent of non-capsulated colonies with cocktails that contain phage 12 or phage 46, we saw that it didn't necessarily mean that we would lose capsule within these mice. And that in some cases, the ones with the purple phage, which as far as we know, don't require capsule for infection, are still good at selecting for non-capsulated bacteria within the gut. Again, pointing to the fact that even sort of our in vitro kind of evolution to sort of map out host factors that are important for phage infection are clearly different in the gut context than they are in a mouse. We can also see other sort of mutants that arise from the mice that are largely driven by phage 38. So these mutants here are either to O antigen or LPS within the bacteria, and they could lead to increased resistance to phage 39, but at the same time render them more susceptible to immune system phagocytosis, which is depicted on the right. And because it will take too much time to get into the details, we could also note as well that particularly for phage 39, that this phage is able to adapt to those mutations that are accrued within Klebsiella, mutating its own receptor binding protein so that it can then broaden its host range and infect these resistant mutants. So in this model that we had ER39 that would infect kind of a wild type Klebsiella within the gut. The bacteria would adapt to that by losing its O antigen, so now that the phage can no longer infect, but then the phage would mutate its receptor binding protein so that it could bind to another receptor on that bacterium and increase its sort of infection cycle. If you remember, this 39 phage was one of the more effective ones in the gut. So we're currently investigating whether or not this evolutionary adaptability is kind of linked to its increased persistence within the gut and its increased effectiveness. So at least with this phage work, I hope to show you that, these phages can suppress the burden and the virulence of Klebsiella within the gut. And one of the major things that came out of it was that in vitro predictions don't match in vivo efficacy. And this is really kind of the genesis of kind of the things that we're currently working on now, at least within the phage program in the lab, which is trying to define what these failure modes are for phage in the gut, be it the heterogeneity and phenotypic state within the gut resident Klebsiella, how spatial niches kind of inform susceptibility or resistance to phage, as well as the impact in the environment on the coevolution between phages and bacteria within the gut. We've also kind of linked up with the Lawrence Berkeley National Lab as part of this larger phage foundry to kind of isolate thousands of phages against clinically relevant pathogens, not just Klebsiella, but also Pseudomonas aeruginosa, Acinetobacter baumannii, as well as the plant pathogen Pseudomonas syringae. We're also doing some, more classical synthetic biology work there, doing protein design of receptor binding proteins so that we can program the host range of some of these phages. And finally, some of this phage work has excited some clinicians that we have here at the University of Chicago, particularly in the urogynecological clinic. And right now we're excited in using some of these Klebsiella as well as some E. coli phages in the case of recurrent urinary tract infections in elderly women to kind of provide an alternative to these women for which kind of antibiotics have failed treatment. So with that, I will acknowledge the folks in the lab, particularly Jack, Tracy, and Ella, whose research I spoke about today. We'd be happy to take any questions.
Fantastic talk. I love it. Especially the second or phase part of the talk. Has huge potential in the future.
Yeah, we're pretty. Yeah.
So kids need to go 3 minutes, so I want to ask him to ask question or comment. first?
Oh, question or comment for Mark?
Yeah.
I don't have any. Yeah, I'm excited for the work. I actually want to talk to you more about these microbial communities because I'm thinking about it in terms of anaerobic digestion, but I won't bore the audience with those questions.
Yeah, no, I think that would be very interesting conversations because especially in the first work that I talked about, what we learned is that like if you look across the aisle, sometimes there are engineers that are in completely different spaces that have developed a lot of techniques that would be kind of useful here. So here, like the industrial Clostridia folks had done a lot of the legwork that we can now go over to this biomedical context. And we're interested in doing some metabolic engineering applications in the gut, which might be very different than sort of, you know, wastewater treatment and those sorts of things. But some of the concepts might be the same or might be second nature, you know, to you being in that.
Yeah, absolutely. Yeah.
So that actually interesting to me because you know I didn't look at the wastewater as a source of the phase, but you're showing that that's the huge source
Oh
of
yeah,
the phase, and then
chock full.
yeah, so that's amazing, and also I'm glad you're working with the Lawrence Berkeley lab because I believe the other Markin, right?
Adam Arkin's part of it. Vivek Mutalik is the
Vivek is
one.
another
Yeah,
person, right? And I
yeah, he's the one that's leading it. Yeah.
I didn't listen to one of the talk. I mean, that's amazing. I glad you're part of that, you know, effort. I really support that should be done, and in that way contribute to the entire the community, you know, working with the phase. That's absolutely brilliant. I see the multiple question from audience. Let me, you know, let me just ask the first question first. The canyon.
I'll jump off and just say thanks again, Taesuk, for the opportunity
Sure.
and so great to hear an update of your research. So
Sure.
I'll
Thank
be
you
in touch
so
with
much.
you. Yeah.
Yeah.
Yeah, thanks,
Bye.
Steve.
See you
See ya.
later. Okay, so the first question from Canyon is, am I correct in the understanding that the goal of both of these studies is a drug in the form of a bacterium or a bacteriophage to treat gut issues like colitis? Would that treatment involve inoculation with the engineered bacteria or phage? Interesting.
You are correct in that. Yeah, I think colitis is a good example. That's some of the things that we've been working on. Metabolic diseases as well. Fatty liver disease is another model we have going in the lab, as well as cancer as well. So there's a fair body of work in the microbiome field that point to the fact that people's microbiomes could affect whether or not they respond to immunotherapies, checkpoint blockade therapies. So we're interested in that respect as well, doing some sort of engineering the microbiome in the context of cancer to make cold tumors hot and those sorts of things.
And then also, can you also elaborate? Because I mean, he also wanted to know, my guess is how you deliver microbes or the phage into the potentially into the human.
Yeah, so both of them would be liquid suspensions that you would drink. So usually with phages, it's a fairly a vial. You drink it. And I've seen people, particularly in Georgia, like Russian Georgia, where phages are kind of a mainstay for antibiotic therapy. There, they grow them in LB or BHI and sort of just drink those lysates. You can buy them over the counter. For the bacterium, it would probably be a similar sort of thing. At least in the SynLogic studies, there would be a bacterial suspension that the patients would drink. It was fairly large in volume, like 250 mils or so. They would drink per day to introduce the bacterium within the gut.
Wonderful. So, the second question by the same person, the Canyon, how long does the phage cocktail evolution take on the ancestor strains, and could you theoretically evolve the phage specifically for each person's gut microbiome? Interesting.
Yeah. So the evolution happens quite quickly. So probably like within 2 or 3 passages or so, we could select for fixed phage variants that occupy 100% of the phage populations that have those mutations. Sometimes, what sometimes there's like, I guess it depends on what the solution is to improve the virulence of the phage. Sometimes there's a single solution, so you always get one SNV in a single protein. Sometimes there are multiple different solutions. So you would sequence a population and you would see that there's a population of phages that exist from there. That kind of depends on the phage and the interaction. And in terms of kind of catering it specifically to the microbiome, that's definitely something that you could do kind of logistically. I think it would be quite difficult sort of to cater each of these different phages for, like, you know, culturing someone's different microbiome and selecting in that case. But I think it is something that is kind of worth pursuing, particularly in the sense, you know, kind of going in again as a synthetic biologist that is fairly design driven. When you look at phages, you know, even these Klebsiella phages, which are relatively well described, we don't know what the majority of the proteins do. So if you would like ask me, like, how would you improve this phage? The real question is we don't really know. So some of the work that we have going on is trying to use evolution as kind of a guide to point to what regions in the phage genome are important for virulence. So that if you passage phage in different environments, in different contexts, and look at what sort of mutations arise and how that affects the virulence of the phage on a specific bacteria, can that point to sort of hotspots within the genome that you can zone in on and say, hey, this is where we should engineer. Maybe we should diversify this protein. Maybe we should take this protein out because it seems to be dispensable, et cetera.
I see. So let me ask one question before we end, because I don't see any more question from audience. So I'm super excited when Arda Markin presented the huge, you know, collection of the phage and then also host, you know, pairing, and that's absolutely brilliant. And I want to see more. And then, but the other thing I want to know is, I'm not the phage guy, so I don't know much about the phage. The phage, if I know, you know, phage is specific, but not necessarily very specific, because that one target genus or species level at the end of the day. I don't know what is the true, but so that's my first question. And then you use a cocktail of phage, and then that is more efficient than just one single phage. That's what I remember from your data. So one potential issue we need to address would be, you know, I call it non-specific or you know, cross, you know, killing, including some potentially beneficial bacteria in the microbiota. So could you kind of comment on how specific phage is, and then how much kind of crosstalk kind of giving some problem, you know, eventually?
Yeah, so I'll say the phages are quite specific. So even in our library of phages that we have, you can kind of bin them into broad host range phage and narrow host range phage. So the narrow host range phage, like in our library of like 80 Klebsiella strains or so, maybe they can infect 2 or 3, and they only infect those that have a shared kind of cell envelope. So maybe they have the same O antigen, the same capsule, et cetera. But then there are other phages that tend to be broader host range. We don't have a single phage that can infect all 80 Klebsiella strains, but we do have some that can infect, say, 50 or 60 or so.
I see.
And then the point is well taken that there could be these off-target effects on the microbiome. Thankfully, kind of Klebsiella, even though it is highly related to E. coli, has a fairly unique cell envelope. So, and since that's going to dictate, you know, how the phages are going to infect, whatever receptors are there on the bacteria, we tend to see very little crosstalk, even between, say, potentially beneficial strains of proteobacteria and Klebsiella. And of course, when you're talking about gram-positives or Bacteroides or other gram-negative members of the microbiota, their cell envelopes are completely distinct, so that there's kind of no effect that we can see there of phages. If anything, the more major concern would be that not a lot of folks are looking at now is the effect of the phages on the host immune system. And there's some data that shows that even just administration of phage can activate innate immune responses that can kind of polarize a mouse at the very least towards inflammation. So the use of the phages in those contexts might not be having these indirect effects on the host that can then either loop back on the microbiome or interact with the immune system in potentially deleterious ways. So I feel like that's an area that has been really underserved and underappreciated, because there's a lot of microbiologists that are only focusing on the microbes for phage therapy. But of course, there's a host there, and understanding those host responses are also important.
That's a very informative, and thank you so much for. So I know, I mean, the team roots work, I mean, those kind of phase work, all the times. I mean, that's now I probably 10 years ago, but that's fascinating. But I just wonder those kind of issues. But that immune response is very interesting as well, and that should be probably addressed in the future anyway.
Yeah.
And I'm not the phage guy, so I basically use recently just Cas9 to just target for the target killing, and then
Yeah.
I believe that some people also, including Team No, also use a Cas9 or Cas system
Yeah.
to introduce the target killing at the end of the day. But still, phage immune triggering probably need to be addressed. I mean, if that is the case. So
Yeah. And I think our understanding of it is right now is we don't know if it's beneficial or deleterious. It could
right.
be that activating the host immune system using phage is actually beneficial to help clear an infection, or it could be the complete other way. That is kind of the current state of understanding
Got
of the
it.
field.
That makes sense. So, the mice system that's CRISPR system, delivered by the liposome is there's no such, no off-target event or probably no immune response. But the problem is the efficiency probably lower than the phage infection, because this is basically fusion between the
Yeah.
bacteria and the liposome. So that is probably one problem I have. So I guess, I mean, at the end of the day, there is no one single solution to one big problem, I guess. So we need to kind of
Yeah.
try all of them, I guess.
Yeah. And usually things that seem simple at first, like with for instance, with the phages, end up being a lot more complicated.
Okay,
What started off as killing a single bacteria
sure.
led to all this evolution nonsense that we now need to deal with.
Absolutely. So I see the one more question. So let's entertain this one. So Jonathan asked the question, how long do engineered bacteria and phage persist in the gut microbiome?
Well, so I'm going to give the disappointing answer of it depends. So it depends on the So on the microbe side, you could assume that as if there's a niche available within the microbiome and your engineered bacteria can engraft, then it's going to stay in that microbiome as long as that niche is occupied. But you could kind of design that. So I gave some examples at the beginning of like using engineered E. coli. So those strains are intentionally kind of neutered so that they can't occupy any niches in the gut so that they're rapidly cleared. But there's other examples where they've even engineered kind of strains of bacteria with novel metabolic properties so that you could create an orthogonal niche within the microbiome so that this bug can persist for a long period of time as long as a mouse is eating a diet. And once you change the diet, then all the bacteria can go away. And then for the phages, for the most part, I'd say the phages will persist in the microbiome so long as they have bacteria to eat. So if they drive a bacterial population to extinction, then the phages will eventually just get flushed out. But as long as that there are bacteria there, you could see this kind of coexistence between a bacterial population and the phages. There's a slight caveat there where there are some phages that can bind to mucus. So there are some models that these phages, even during ingestion, they'll bind to the mucus and stay there for a period of time and kind of act as this, defense system that we can have. So maybe those phages can exist for a little bit longer, even in the absence of a bacterial host. But I'd say for the most part, if the host is there, the phages will be there.
Wonderful. So I want to comment on that question. So that's absolutely great answers. One thing I wanted to add is, the regulatory agency not necessarily want the engineered bacteria, we also call it GMO, stay in the gut too long time, because so some people also worry about those GMO potentially become bad guy in the gut. So that's one of my NIH grants actually initially criticized, because we wanted to make the bacteria colonize longer, and then we kind of now saying they will just stay there very short time and then basically wash you away, and then that is like the better way in terms of those kind of potential problem, also biocontainment issue, etc. And then for the company perspective, staying less means everyday consumption of the probiotic or whatever, you know, bacteria, and then that's better for the company, you know, revenue in some sense. So there are
Yeah.
multiple factors you need to consider, and that's one thing I want to add to the answer.
Yeah. No, thanks for that. And then I feel like I should always mention biocontainment when you're talking about engineered bacteria, and I
Yeah.
forgot. Thank you for doing so.
All right, so I guess I don't see any more question. Also, I realize I need to catch my flight very soon.
Oh
After
boy.
probably 4 hours of sleep. So let's close. Okay, so thank you all for joining and staying today. Now my time is after 1 a.m. And as I mentioned earlier, after 4 hours of sleep, I need to leave. I need to leave my parents' home in Korea to go back to San Francisco to take my booked flight from San Francisco to Denmark and then Sweden. So I probably need to close now. So we'll meet again on
Yeah.
August 29, Thursday, the same time and same Zoom link with me meeting you from Sweden, Europe. We'll have the Professor Avi Schroeder at Technion, Israel Institute of Technology, and the President-elect of Controlled Release Society, and Dr. Brian Huang at Harvard Medical School. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us, and I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Just 1 second. Okay, stop.